Welcome, and thank you for visiting my video. My name is Mizuki Yoshino. I am a 4-year undergraduate student in the DGTalAquaLab. In this video, I will present the result of my research in the form of a poster presentation. As an undergraduate student, I am working on the development of an individual-based model which will serve as a tool for culture management of a tiny crustacean called copepod. And I started this endeavor by developing a model that simulates the effect of salinity on the reproduction of cyclopoid copepod Aquaculture roy. This poster present is the first step and was presented in Japanese during the annual meeting of the Plankton Society of Japan in mid-September 2021. To start, it is important to understand the importance of aquaculture in our current society. To keep up with the increasing human population, seafood production via aquaculture has increased in the last 3 decades. It has been estimated that the seafood production via aquaculture represented 50% of the total production in 2019 and should reach about 60% 30 years from now. This high demand led to finding the most appropriate live feed for fishes. In aquaculture, fish are fed a variety of food to enable them to live and grow. The food depends on the developmental stage of the fish. Artemia, copepod, rotifer are commonly used to feed the fish larvae. Compared to rotifer and Artemia, copepod are more nutritious and have great potential for larva predators. However, copepod is not commonly used as live feed in Japan due to the difficulty in production and management. In aquaculture, mathematical models are helpful for reaching production goal. They can accurately predict animal growth, estimate energy requirement, and calculate dairy rations. And there are practical applications of individual-based model for a variety of fishes in the literature, but not for copepod. We therefore aim to create a genetic simulation model that helps in establishing copepod mass culture through determinations of optimal culture conditions for growth and reproduction, and define appropriate harvest time. For the development of the model, we focused on a species used as live feed in Taiwan, the cyclopoid copepod Aquaculture roy. Dr. Pan from the National Taiwan Ocean University successfully mass cultured this species and collected several data required for the model development. Our model is an individual-based model that simulates the reproductive cycle of an egg-bearing copepod, similar to the one of Dur et al. 2009. Females enter this cycle after molting from the last juvenile stage C5 and stay in this cycle until their death, defined by their survival probability and their longevity. The cycle integrates 2 main processes: the spawning of a clutch site and the hatching of the egg. The between hatching and spawning is defined as latency time, and between spawning and hatching as embryonic development time. The female status is also conditioned by these processes. And we use the Mobydick platform to create a model for Aquaculture roy. The reproductive cycle was represented by the following flowchart. This flowchart represents the scheduling of the model processes used to define the reproductive cycle of Aquaculture roy. And you can notice that the conditional task, spawning, and hatching require latency time and embryonic development time to reach the threshold value for their execution. These thresholds were shown to be salinity dependent as well as the clutch size and the female mortality. We therefore spent some time parameterizing these effects of salinity on the reproductive traits. For EDT, LT, and mortality, we fit data obtained in the lab and from the literature with 7 commonly used development functions. The equation of Yamakawa and Matsuda provide the best result in terms of goodness of fit, and minimum and max values that can be associated with the range of trellis of Apocyclops roy. For the clutch size, we modified the Parker equation, which was used in the older paper to describe the effect of salinity on the clutch size. Those functions were implemented in the model and run some simulations for 15 days with 1,000 females under 8 different salinities ranging from 0 to 35. The output from those runs performed on Moblick are presented here. The top graph presents the cumulated egg production of each time steps. The bottom graph presents cumulated offspring production of each time steps. For both, the highest production was observed for salinities between 20 to 25. Besides, the lowest salinity did not suit Apocyclops roy. When looking at the relation between the cumulated production after 15 days and the salinities, we can first see differences in the number of eggs and larvae, which are due to the incorporation of the hatching success rate into the model. Also, we can clearly see that the production increases with salinity up until 20 to 25 and decreases for higher salinities. Therefore, we can conclude that the best salinity for Apocyclops roy reproduction is between 20 to 25. With the current model, we could define the best salinity condition for reproduction and estimated the total production at such optimal condition. This is a small but important step toward the creation of model that can help defining the optimal culture conditions for Apocyclops roy. As we introduce other environmental factors in the model in the future, we will be able to define the other conditions such as optimal temperature, optimal food conditions, and so on. So stay tuned for more news on the progress of our model. If you are interested in this lab, please have a look at the website. You will find many information, including the contact of the PI, Dr. Duragile. And for more videos, don't forget to subscribe. Cheers!